Thank you very much, Peter. I'm very much touched, but I wasn't sure, do you want me to talk about the arts or the science?
Whatever.
All right. So my presentation is focused on the research that I've been doing and the method that I've developed over the period of time. But since there are so many young scientists here, I thought it would be appropriate to just talk about my own Werdegang, which means that my own trajectory from very young to old. Part of it has already been taken over by Peter, but I will give another version of it. And then I'm going to talk about what it is all about. What is structural biology? Why do we even bother? And then I'm going to go into my particular method and how it has developed. So, Szeged, where I come from, it's actually not so far away from here, in the middle of Germany. And this is the picture where I'm presented for the first time in 1940. And this is the house, my parents' house, a very big, stately house. And just on the left-hand side, it's very difficult to see, but that's the veranda under which I did my science experiments as an 8-year-old. And that's where I built little contraptions and collected bottles and so forth. So I always was carried by an immense curiosity, and I did science experiments before I knew what science was. And let's see. And then, because study of physics was sort of a natural idea for me, I went to Freiburg, University of Freiburg, and studied there. And then afterwards, I went to Munich. And all these choices are really carried completely. They have nothing to do with science. I really wanted to be in Munich, and some people went to Munich, so I followed them. And that's how I got there. And that's how I wound up in the laboratory of Walter Hoppe, he was an X-ray crystallographer who developed an interest in electron microscopy. And so I was at a lab that really brought both concepts directly together, both theoretical foundations. And I was in a certain way lucky to wind up there. And then, this has already been mentioned, I was lucky to got nominated and selected for the Harkness Fellowship, which was really sort of an unspecific fellowship that encouraged me to make contacts with different labs and travel around. So one of the first places was the Jet Propulsion Laboratory. And I developed very early on an interest in digital image analysis, in electron microscopy. And at the time, Jet Propulsion Laboratory was the first, #1 place in the world with equipment and software development in image processing. And from there, I went to a lab in UC Berkeley with Robert Glaser, and it happened to be exactly the time when he explored the benefits of going to low temperature in electron microscopy to keep the radiation damage in control. So it was a very crucial time for me to wind up there, and I made many contacts that were very valuable later in my career. And then the third step was at Cornell University. And I always continued to develop programs. And then the next step was the Cavendish Laboratory, which is a very famous laboratory and it has a distinguished history of experimental science, all related to nuclear science. And I spent there for 2.5 years. And this is where, Ernest Rutherford was one of the directors, and he discovered the nucleus, and he was appointed as Cavendish Professor in 1919. And this is a very unique picture. This is the courtyard of the Cavendish Lab, and you see a barrack. What is the barrack known for? Well, it is the very beginning of the Laboratory of Molecular Biology. This is where Max Perutz did his first protein crystallography. So later on, the lab moved out to Addenbrooke's Hospital, and now, of course, it's a unit all by itself. And here we see Max Perutz and John Kendrew, his son, his student, with the model of hemoglobin, which is the protein that carries oxygen in our blood cells. And they received the 1962 Nobel Prize in Chemistry. And then the next step brought me to Albany. And this is where I had the leisure to develop the ideas that I had in my mind of the single particle reconstruction. And I used ribosome in order to make this happen. So this was a very, very important time for my career. And I was left alone. I didn't have a lot of teaching obligations. I was a senior research scientist employed by the New York State Department of Health, and that really gave me the time that I needed to invest in these ideas. And then New York, of course, the move in 2008 brought me into an environment with much more contacts, with much more access to students, and this is where I really could develop the ideas even further. So now, what is this all about? Why is it so important? Well, the cell is a chemical factory, and there are so many things going on. All life processes, condensed molecules are very acting very closely together, and it's very difficult to see what they are doing unless you take the molecules out of their natural context. But first of all, to understand how molecules interact in healthy or sick organisms, we need to know their atomic structure, position, identity of each atom, and the most frequent elements: carbon, oxygen, hydrogen, phosphorus, nitrogen, calcium, as most of the rest of the periodic table is not being used very much. Here's an example of human protein. I already showed it before, hemoglobin. It's very difficult to make sense of this, okay? These are the atoms. 10,000 atoms of hydrogen, carbon, nitrogen, oxygen, and sulfur, plus 4 atoms of iron. And they are there somehow held in this position. There are two ways of presenting it. One is the very nonsensical way that I did before, and the other one is the so-called ribbon diagram. That shows how the atoms are bundled and connected along helices. Now, where do these helices come from? Well, first we have to know that the protein molecules in our cell are made by little molecular machines called ribosomes. And the ribosomes string amino acids together according to instructions in the genetic blueprint, the DNA. So this is where the central dogma comes in. There's the DNA containing instructions. These instructions are copied into mRNA in a process called transcription. The mRNA finds its way to a place in the cell where it can ask a ribosome to make the actual product, the polypeptide. Okay, so this is a three-stage process, and that explains why the atoms are actually arranged in a string-like fashion. So there is some kind of an underlying linear structure behind it, and that linear structure is the polypeptide. So protein structure is the primary structure, and then the secondary structure, the primary structure is arranged in some way in alpha helices, beta sheets, and then comes the tertiary structure. This is all folded together, and it comes to a next step of a quaternary structure, and that's the actual protein that's doing its work. And so a protein is a chain of amino acids, a polypeptide, bunched together like a coiled up chain of Christmas ornaments. That's for me the sort of idea behind it, where each of these little candles is an amino acid. And you bunch them all together, like you do maybe after Christmas, you just, in order to put it somewhere in a corner. So in a healthy organism, all these processes go smoothly. All molecules in the cell are made correctly from the right genetic blueprints. The immune system protects the body from hostile organisms like viruses or pathogenic bacteria. And many diseases are caused by infections. Viruses, bacteria, parasites invade the body and cause havoc. And to fight infectious diseases, we need to know how the life functions of these invaders differ from ours on the molecular level. And many diseases are caused by mutations. Genetic blueprint contains an error, causing a wrong amino acid to be put in a critical place in the protein, which causes it to malfunction, compromise the organism's health. And this can also affect the immunity of the organism, its ability to defend itself against infectious diseases. So today's medicine is guided by the knowledge of the way molecules are built and the way they act in concert. And with that knowledge, effective drugs can be developed, small molecules that are designed to bind to target molecule and thereby change its behavior. And this is, in a nutshell, is the definition of molecular medicine. So this is what we need, but how do we get this structure? Well, I told you about before. There's a crowdedness in the cell. It makes it very difficult to get hold of single molecules. And so we need to extract the molecules from the cell, purify them in vitro, and then we can study them in isolation. And to look at molecules, can we use the light microscope? No, not at all. They're too small, even much smaller than the wavelength of the visible light. So the visualization, we use the electron microscope. Well, we can look at see the electron microscope in close parallel to the light microscope. We have lenses, but these are not made out of glass. They are magnetic fields. But the image formation is exactly the same way as with the light microscope. And a very big difference is that the resolution is much better than light microscopy. So the resolution is smaller than 0.1 nanometers. And the first microscope was created in 1931 by Ernst Ruska and Max Knoll. Ernst Ruska received the Nobel Prize for this in the 80s, I believe in 86. And this is, I can only reinforce what Dan Shechtman told you before. If you have a bright idea, put it in a notebook. Don't put it in a computer. Put it in a notebook. These things stay around, for a very long time. And there will be a time when you can pull it out and say, I was there, I did it. So the transmission electron microscope is basically sort of vertical in design. There's a place up here where electrons are created in the source and accelerated to very high speed, and they move in high vacuum. So the column has to be on high vacuum, because otherwise the electrons get stuck very, very quickly. And here is an illumination system that focuses the beam on the sample. The sample is very close to the objective lens. The objective lens is the most important lens in the microscope. And in here, over here, is an intermediate image formed, which is further projected and magnified by the projector lenses. And down here, and this is a very old cartoon, which still showed plate cameras and a fluorescent screen. This is no longer used. Instead, we have cameras, electronic cameras, and we're not sitting by the microscope, but somewhere else at a workstation. So we are all talking about cryo-EM. Where does the cryo comes from? Well, that comes from a discovery by Jacques Dubochet in 1981. He discovered that by freezing molecules very, very fast, you get a state of the water that is unlike other states that had been discovered before. It's vitreous. So the water stays essentially at a glass-like state without expansion, without formation of crystals. And if you use this kind of method of cooling, then the molecule stays intact and is not being destroyed in the process. So it was a very important discovery in 1981. And this is essentially the process. You pipette the sample onto the grid, and then there is a step of blotting so that you get the solution into the right kind of thickness, and then it's very rapidly plunged into the cryogen. This is the kind of picture that you see. It's very grainy because we want to use very low exposure so we don't inflict too much radiation damage on the molecule. So what is three-dimensional imaging about? Well, first of all, the transmission electron microscope creates two-dimensional projections. So we don't really get a three-dimensional image. In order to get the three-dimensional image, we have to take an entire series of pictures in different directions and later combine them. And there's an important theorem, a mathematical theorem, which tells you how to do this in practice. So my idea was the single particle methods. A single particle means that I don't assume that the molecules are somehow in a crystal, but rather I make use of the fact that the molecules are simply they're floating in liquid. They're under all kinds of different orientations, and so I make use of this fact and try to combine information from molecules that are exposed in the same orientation and have the same structure. So in order to make this all happen, to combine all this information from different molecules that exist in solution, I had to develop a very large number of programs because these solutions didn't exist at the time. So I had to put myself together a workbench of programs, something like 400 programs that all were able to work together in concert. So there are several problems had to be solved. I mean, the most important problem is, of course, to find the projection angles after the fact. We know that it is the same molecule, but it shows different orientations. And how do we find these orientations after the fact? We have not tilted the molecules, they have tilted themselves. And so this is finding projection angles, aligning the images, sorting and classifying the images, and reconstructing them in 3 dimensions in an unknown geometry. So these are all challenges that need to be solved by software. So the first, when this technology came together with the cryo sample preparation, this was really a eureka event where we saw for the first time 3-dimensional images of molecules that had not been seen before because they could not be crystallized for X-ray crystallography. So all these pictures here had never been seen before. And so it was an incredible gratification for me to see that the technology that I had developed made this possible. But these, you can see that these images, nice as they look, are still at very low resolution. And in fact, at that time, I did not dream that it would ever get into atomic resolution. I thought this is a very useful contribution to science because we see molecules that cannot be seen with X-ray, or sometimes we can see molecules in different conformational states that could not be seen before because they were squeezed in the crystal. But we never dreamt that we one day got to a place that we are now. So in the year 2000, we had this, the first FEI TechNI instrument on the American continent was in Albany in my lab. And FEI at the time, now it's Thermo Fisher meanwhile, FEI made a very bold step and invested in the technology which brought together the very complicated electron optics and the computer control into one box, and they called it TechNI. So this was the first completely integrated microscope at that time. So with the technology that had been brought together, I was now able to look at the ribosome in the different stages of its process. We were able to map out the entire elongation cycle. One elongation cycle is used for adding one amino acid to the growing polypeptide chain. So we were the first ones to be able to map this out. So the ribosome is very curious in its entire process because I'm a physicist. I didn't know what a ribosome was. I had no idea whatsoever. It was somehow something, somebody showed me a picture of a ribosome very early on and said, is this would this be useful to demonstrate your technique? And I said, oh, it looked wonderful and so forth. But I had no idea what the ribosome was doing. And then a few years afterwards, it dawned on me that I could really make a contribution to biology. And then I learned in the process what the ribosome was doing and its central role in the cell. So then I became partially methods developer and partially I became a very specialized biologist, only translation, okay. So somewhere around after 2000, something like a workflow developed. And meanwhile, that workflow goes all the way from sample to atomic structure, from protein purification, and then there's first a step of negative staining just to see whether the molecules show up at all and whether we really find what we expect to do to get. And then we go to cryo, and then take thousands of pictures, and then comes a very important point of three-dimensional classification, because we found out that it's possible to extract different molecules all at the same time from the same sample. So if we image something, we might get a whole surprise. We might get 10 different structures that all show a molecule in different states or different ligand binding states and so forth. So there's a before and after. It's a before and after 2012. Before 2012, it was very, very hard to get even close to something like 5 angstrom resolution or 0.5 nanometers. And then in 2012, 3 different companies came out with single electron detecting cameras. And these are so sensitive and have very high resolution that they allowed the jump to atomic resolution later on. So it was a milestone year. And so if you compare the literature, if you look up anything that was published after 2012, 2013, all of a sudden the world is completely different. You know, you see thousands and thousands of structures that were obtained this way. And you can show, I can show this by comparison with what I showed you before, with ribosome recorded on film versus ribosome recorded on one of those direct electron detection cameras. You see very high contrast, which means that when you get very high contrast, since all these processes involve correlation functions and three-dimensional classification, they all involve comparison between molecules. So if you get high contrast, then this comparison gets you very high resolution, alignment, and so forth. So, so this step here made it possible to get into a range where structures could actually be used for developing drugs. So they got into a range of resolution that was similar to the one achieved by X-ray crystallography. And these are sort of the fully integrated TechNice series. Nowadays, it's spread out all over the world. And this is what the inside looks like. It's an incredibly complicated instrument, $6 million, and then plus maybe the $4 million or 10 that you have to spend in order to prepare the room for it. So the first time this had an impact for my own research was in 2016, where we achieved the 2.5 angstrom structure of the ribosome, and it's really stunning to see that we can recognize individual ions. We can see base pairing. We see the signatures of amino acids and bases. And here, just to show you this impact of this, show you this three-dimensional image. And from a three-dimensional image, one can then infer the atomic structure when the resolution is good enough. This false color representation shows different resolutions. When it gets very purple, is the highest resolution at 2.3. We are just going through this like through a dough. So I show you a few examples of structures that I've been directly involved with later at Columbia in various collaborations. And here's a ryanodine receptor, which is really the most incredible achievement. It's a very, very large structure that is instrumental in muscle contraction and the beating of the heart. And so we got this to atomic resolution in 2016. And here's an example of cystic fibrosis of the chloride channel, which is responsible for the degenerations that leads to this terrible disease. And here's the atomic structure, and it shows the missense mutations that have all been mapped on the receptor. You see the number of different diseases that are all produced by mutations here, and it was for the first time possible to map these into the atomic structure. And I was so moved in 2017 when I received this message. Dr. Frank, I want to add the chorus of congratulations that you have no doubt received since the Nobel was announced. I have special reason to send you thank you as well. My 7-year-old grandson Jackson was diagnosed with CF as a newborn in 2010. I have attached news that we received this week regarding his 10 months on combination therapy. I just wanted to share this as a small expression of my and Jackson's gratitude. It was unbelievable to see this, because I had not even made this connection, but he made this connection. He thought, this technology had made it possible to get to this kind of resolution. And then, so that brings me to COVID. The technology really has very strongly contributed to the vaccine development and development of assays to find the antibody positions. And this is this very significant anecdote. Just 2 weeks after receiving the genome sequence of the virus from Chinese researchers, the team had designed and produced samples of the stabilized spike protein. It took 12 more days to reconstruct a 3D atomic scale map called the molecular structure of the spike protein and submit it to the manuscript of Science, which expedited its peer review process. So within a very short time, we had a science paper, and on the basis of the science paper, much of the subsequent research could be done. And I'd also like to mention Katalin Kariko, who developed the mRNA vaccine not after this structure was discovered, but after a very similar virus was analyzed that had a similar attack mechanism 2 years before. So this is one of the amazing fortuitous facts in the history of this, that just by this historical accident, we already knew what was coming there, okay? So we had a more benign virus. We had information about a more benign virus from 2 years before that enabled us to do the mRNA vaccine development for the much more deadly virus that came afterwards. And that brings me up to the time when these amazing things happened in my life and in the life of Carol and me. And here we are. And I have to thank Carol for sticking all through this years and years of frustrating time with me. And here we are on 2018 on top of the Black Building. I just wanted to tell you, go to my lab site, the website of my lab, and you find something that is called Nobel 360. And Nobel 360 is a special link that links you to a whole 360-degree environment in my lab and outside of my lab that was taken by a photographer that visited me afterwards. So if you want to see a little bit more of my lab, then you go there. And then, as Peter already mentioned, Frank's Fiction is a site where you can access lots of things, lots of ideas that are completely unrelated to science, but nevertheless, they are sort of part of me. Okay, thank you.
So now this talk is also open for questions. So anybody has any questions? There was one student who came to me that couldn't ask a question, is here.
Hello. Do you mind answering, how do you see the role of AI evolving in structural biology and image reconstruction?
I think AI overall is immensely overrated and is also misunderstood. The role is misunderstood. Science is experimental science and not simulation. AI, such as AlphaFold, is sitting on or has exploited the wisdom of decades of experimental research. More than 150,000 structures are in the PDB, and so all that knowledge is based on this. Outside of that knowledge, there's nothing. Okay, AI cannot come up with structures that have not already been explored. So it's somehow useful to give you hints on how structures might be folded. It can give you a whole smorgasbord of solutions, but you have to do experimental research in order to validate this. And there are some people who actually think, after AlphaFold, what do we need experimental science for? And this is this is an unbelievable misunderstanding of science.
Thank you. Other questions?
Thank you.
We still have time for a question.
Thank you, Professor Frank, for this great talk. So throughout your talk, you mentioned your education, you mentioned the state-of-the-art technology and equipment that you use, and also your connections and networking. Which of the three do you think was most important for your success?
What was the last one? I didn't hear that.
So you mentioned throughout your talk, your education, your state-of-the-art equipment, and connections and networking.
Oh, connection networking.
Yes.
Yeah.
Which of the three do you think was most important for your success?
I wouldn't know. I mean, I think without connection, you still get it done, but it gets done slower. It would never be the most important thing because you need a guideline. You need an idea of what to do. And the equipment is similarly. You assemble the equipment, or you get hold of the equipment when you have an idea to that you want to realize.
Thank you. And one more question, if the students. One more.
So what skills or mindset do you believe are the most essential for the next generation's scientists?
What tool?
What kind of skills or
What
mindset?
skills? Well, I think just because of the invasion of half-truths and non-truths, critical ability to sort through this morass of the internet is very important now. So I never thought that we get to the stage where the discrimination of what is important, what is true and what's not true, has become the most important thing. Because you have really, in this situation now, which is completely unlike my situation when I started, you have all of a sudden everything on your fingertips. You know, you can, if you have the right ideas and creativity, you can do unbelievable things done very, very fast. But you really need to be sure that you're on the right track, that you're not following paths that are completely irrelevant.
And I think the second part of her question was that what kind of personal skills for a young scientist could be in the future?
Oh, personal skills. Personal skills is really to be open. I call it peripheral vision. To be open to things that are completely outside of your plan or your initial idea. So sometimes a solution come across in a very unexpected way, and so keep your eyes open for the completely unexpected. And very often, negative results in experiments can have a very powerful message. In my whole career, it happens a number of times when something totally unexpected happened, and then there was a reason, and the whole and the results sort of changed my direction of research in a very positive way.